Hello, everybody. I am the Dr. Bennett, and today I'm going to be going over Khan Academy's MCAT Biomolecules DNA questions. So, the practice set of DNA questions. There's 11 problems here, and let's get started with the first one. The lagging strand of a DNA molecule undergoing replication reads from 3' to 5', and they give a sequence. What is the code of the DNA that is the template for the complementary leading strand of this segment? Okay, so we're looking for the template of the complementary leading strand. So the complementary leading strand obviously is going to be 5' to 3' complementary to this. So where you see where there's a C, you're going to see a G, an A, you're going to see a T. However, they're looking for the template for the complementary strand. So wouldn't the template just be this, what they're showing us? Where that is one of the answers, so I'm going to check that. Boom. Okay, it's right. So yeah, that's what they're doing. That was weird wording. Tip my hat to you, Sal. But yeah, that's so they're looking for the template for the complementary strand. Remember, the complementary strand, whether you're talking about leading or lagging, they're complementary to each other. But if you want to know what's the template for one of them, it's the other one. Okay, next question. So, at what point during normal DNA replication is genetic material lost from the telomeres? Okay, so telomeres, I know they're at the end of chromatids. What do we got here? So we got the attachment of DNA polymerase to the leading strand. That doesn't seem likely. The joining of adjacent Okazaki fragments. That could be it because that's about DNA replication, synthesizing new DNA. The enzymatic action of telomerase. Ooh, that's a trick question because telomerase, I think, increases telomere length, doesn't decrease it. And the question is looking for at what point during normal DNA replication is genetic material lost from the telomeres? Okay, and then the final answer choice is unzipping by DNA helicase. So the only one that makes sense is joining of Okazaki fragments. And sure enough, that's right. Okay, let's look at the hints real quick, though. Okay, telomeres are the caps at either end of a chromatid, which protects the DNA from deterioration. Okay, cool. The shortening of telomeres is due to end replication problem. This problem arises from the fact that DNA polymerase synthesizes DNA only in the 5' to 3' direction. Right, and that leads to you having to do the Okazaki fragments. Okay, that's the next hint. Okazaki fragments are pieces of newly synthesized DNA along the lagging DNA strand separated by RNA primers. The final RNA primer at the terminal end of the new strand cannot be converted to DNA by DNA polymerase and joined to the strand by DNA ligase because there's nowhere for these enzymes to bind. As such, this last primer is degraded. Oh, okay. And thus, each successive DNA copy is slightly shorter than the last. That makes a lot of sense. Okay, so joining of Okazaki fragments, that is going to decrease telomere length over time. All right, third question. Which association between complementary bases would require the most energy to break, meaning it has the highest bond dissociation energy? Is it AU, GC, all are equal, or AT? I'm definitely going to say GC. And if you think back to your bases and how they work, you have pyrimidines and purines. And if you look at how they actually bind together, then you'll see that when you have GC binding together, you get 3 hydrogen bonds instead of 2, which is what you would get from AT or AU. And sure enough, yes, GC is correct. All right. Question number 4. The antibiotic doxycycline is known to bind and inhibit a particular ribosomal subunit to inhibit bacterial proliferation. How might this affect the 80S ribosome in human cells versus the 70S subunit in prokaryotes, and why? Okay, so let's look at the first answer. Doxycycline would inhibit ribosome function by binding the 30S subunit because both humans and prokaryotes share the 30S subunit. Okay, if I remember correctly, they're different from each other. And one thing I remember specifically from my biochemistry lab is that the Svedberg units of each individual subunit do not add up to the Svedberg units of the actual organelle. So that one doesn't really make any sense because we take doxycycline. So it shouldn't inhibit our ribosome function. Okay, second answer choice. Doxycycline would not affect human ribosomes because the human ribosomes are made up of 30S and 50S subunits, while prokaryotes are made up of 30S and 40S subunits. Okay, so here we go. This is the thing that my biochem lab helped me with. So I knew they were gonna do this to us. So they're saying humans are made up of 30S and 50S, and prokaryotes are 30S and 40S. When you add those up, you get 80S and 70S, which are the actual Svedberg sizes of our ribosomes. However, I know that that's not the case. I know that the sum of the subunits, Svedberg units, is larger than the actual when you do the whole thing together. Because remember, Svedberg is just about sedimentation, how it settles out when you centrifuge a bunch of lysate, cell lysate. Okay, third choice. Doxycycline would not affect human ribosomes because human ribosomes are made up of 60S and 40S subunits, while prokaryotic ribosomes are made up of 50S and 30S subunits. That sounds very familiar. I'm going to click it real quick, but I'm going to read the last answer. Doxycycline would inhibit human ribosome function by binding. Okay, I don't think it's inhibiting human function, so I can get rid of that one as well. I'm going to check. Yes. And in fact, the right answer was doxycycline would not affect human ribosomes because human ribosomes are made up of 60S and 40S subunits, while prokaryotic ribosomes are made up of 50S and 30S subunits. Awesome. Okay, question number 5. A point mutation within what functional division of a DNA sequence would be most likely to ultimately result in the production of proteins which differ from the non-mutated form by only a single amino acid? So this is either like an insertion or deletion. Oh, no. This would be a, I mean, it's a point mutation. I feel like there's another word for it when you just flip-flop. You take out one, you put in another one. But regardless, let's see the answer choices. Centromeres, exons, introns, telomeres. Okay, I know centromeres and telomeres are going to be right out. That's not going to change the protein at all. And once again, they said the production of proteins, so introns are right out. It's got to be exons because introns are spliced out before, with the exception of some alternative splicing, in which case little parts of the introns might be kept in. Introns are spliced out. Exons are then connected together and sent to the ribosomes to be translated into amino acid sequences. All right, question 6. In what direction is DNA synthesized when catalyzed by either DNA polymerase or reverse transcriptase? Okay, so I am notoriously bad about remembering the directions of DNA synthesis and DNA like reading. Like what 3' to 5', which direction does it go? So I'm pretty sure synthesis of DNA from DNA polymerase happens from 5' to 3'. It reads it from 3' to 5'. That is in my head. My girlfriend constantly tries to drill that into my head, and I refuse to learn it. But reverse transcriptase, that's a bit odd. You're going from RNA to DNA. I'm just going to say that DNA always gets synthesized from 5' to 3'. So when I look at my answer choices, I see both DNA polymerase and reverse transcriptase catalyze the synthesis of DNA in the 5' to 3' direction. I'm going to go with that. Yay, it was right. Awesome. Okay, let's look at the hints real quick, though, because I want to make sure I'm thinking correctly. Okay, during both transcription and DNA replication, DNA templates are read from 3' to 5'. Cool. Reverse transcriptase is reverse in that it catalyzes the synthesis of DNA from RNA. Reverse transcriptase still reads its template from 3' to 5'. So both DNA polymerase and reverse transcriptase catalyze the synthesis of DNA in the 5' to 3' direction. Awesome. Always 5' to 3' for synthesis, and then I suppose always reading from 3' to 5'. Okay, question number 7. An individual presenting with a mysterious disease affecting his connective tissues is found to have numerous defects in multiple regulatory proteins. If these proteins are characterized by highly abnormal patterns of glycosylation, to what structure might the patient's disease most likely be attributed? Okay, it's affecting connective tissues. Regulatory proteins. Okay, I don't think that first sentence is really that meaningful. They said the proteins are characterized by by highly abnormal patterns of glycosylation. Okay, so let's look at the answers. Mitochondria, that one doesn't seem right. Golgi apparatus, ooh, okay, well, hold on to that. Lysosome, no. Ribosome, no. Okay, so the only one that makes sense is Golgi apparatus. And sure enough, in my head, I remembered that, obviously the Golgi apparatus, we always think about it. It's like the FedEx Center. It's the UPS. It's sending out. It's not the U.S. Postal Service because they suck. And it's sending out the packages everywhere. It has to put labels on them. It has to do modifications to the proteins before they're sent out to where they need to go. So I would imagine that's where glycosylation happens. If my computer would let me click the answer. Yes, and it was right. Let's look at the hints real quick. I'm pretty confident on this, but I just want to check them out to see what they have to say. Okay, mitochondria are the cytocellular respiration. Yeah. Lysosomes, yeah. Okay, ribosome, give me some stuff about the Golgi apparatus. What? Come on, Sal. The only hint about the Golgi apparatus, the only info you're going to give me is that this patient may have a disorder which is affecting the function of their Golgi apparatus. I want to know more. I want to know why is the Golgi apparatus wrong. But you know what? That's okay. That's for me to go back and learn later, and for all of you to go back and learn later. These are just question answering tips and logic and figuring out MCAT questions. Okay, question number 8. Pyrimidine dimers resulting from UV light damage to DNA are removed from sequences via the action of what class of enzymes? Okay, removed. So as soon as I see removed, I think endonucleases. Let's look at their answers. Polymerases. I don't think, no. I mean, they might be involved later, right, in making new DNA. Ligases. Yeah, they're going to be involved too. Helicases, they'll be involved too. Endonucleases, the fourth answer. Okay. So let's check it, and it's correct. Let's see if Sal gives us a little more information about endonucleases. So he talks about helicases, talks about polymerases, ligases. Okay, here we go. The fourth hint. Endonucleases cleave specific sequences of DNA, such as pyrimidine dimers, resulting from UV light damage to DNA. Right, so endonucleases, their job, I'm pretty sure, is to to cleave, to chop up the bad bits, and then let the rest of the stuff come in and fix it. Such as a pyrimidine dimer. Okay, question number 9. What is the functional relationship between codons and amino acids? Okay, what do their answers look like? Many different codons may code for the same amino acid. Okay, they're talking about the codon chart. So that first answer, yeah, that actually is right. Many different codons. It's the redundancy thing, right? You can have several different, like 4 different codons all code for alanine or something. I'm pretty sure that's right. Let's look at the rest of the answers. Codons and amino acids randomly associate... No, hell no. A single codon may code for many different amino acids. That is not true. A given amino acid is only coded for by a single codon. Okay, so the correct answer is many different codons may code for the same amino acid. And it's correct. Yay. All right, question number 10. Where within... Why did I pronounce it that way? I chose the inflection of where within the eukaryotic cell when it was supposed to be where within a eukaryotic cell might a drug which exclusively binds to tRNA binding sites exert its effects? Okay, tRNA binding sites, I'm thinking ribosomes, right? Probably. So let's see what the answers are. The nucleus, probably not, but tRNA comes from the nucleus. The mitochondrial inner membrane, no, that's out. The lysosome, no. The cytosol, okay. Even though I'm pretty sure most ribosomes are on the rough endoplasmic reticulum, there are ribosomes in the cytosol. Therefore, I'll bet that's the answer they want. And it is. Awesome. Let's look at that step-by-step solution real quick. Okay, tRNA binds to ribosomes to be used in translation. Yeah. Translation occurs in ribosomes. Yeah. Transcription occurs in the nucleus. Blah, blah, blah. Come on, give me something here, Sal. Okay, ribosomes are abundant in the cytosol and on the rough endoplasmic reticulum. Translation occurs within the cytosol. Okay, cool. Yeah, and I guess in the rough endoplasmic reticulum, it's on like the outside, right? So you're technically still in the cytosol, even though you're on the rough endoplasmic reticulum. Sorry, I'm talking real quick, people. Question number 11. Hopefully I don't trip over my words. I'm just getting over being sick, and pre-Dr. Bennett is feeling great. Okay, what constitutes the phosphodiester bonds in DNA? Is it covalently linked nucleotides, hydrogen bonds between complementary strands, covalently linked amino acids, hydrogen bonds between nucleosides? Well, first off, I know it's a covalent bond, so that gets rid of two of the answers and leaves me with either covalently linked nucleotides or covalently linked amino acids. I know there's no amino acids in DNA, and I also know what a nucleotide is, so I'm going to choose that, and it's correct. Covalently linked nucleotides. You have a bunch of nucleotides all in a row, you create a strand of DNA. Awesome. Strong work, gang.